Okay, hello everyone. Welcome to the Brown University Cancer Center Invited Research Seminar Series. Thank you for joining us this afternoon. Please put your Zoom on mute to cut out the background noise. It's great to have Dr. Nellie Polyak from Dana-Farber Cancer Institute with us today. Dr. Polyak is Professor of Medicine and Medical Oncology at the Dana-Farber Cancer Institute and Harvard Medical School, where she's also co-leader of the Cancer Cell Biology program at the Dana-Farber Harvard Cancer Center. Dr. Polyak earned her MD degree in Hungary and her PhD at Cornell University and Memorial Sloan Kettering Cancer Center. As a graduate student with Joan Massaguet, she discovered the cell cycle inhibitor P27. She completed postdoctoral studies with Bert Vogelstein at Johns Hopkins before joining the faculty at Dana-Farber in 1998. Nellie is a leader in the field of breast cancer research, having unraveled tumor heterogeneity and tumor evolution. She has gained insights from studying polyclonal tumors and how they progress and metastasize, and has investigated how clonal diversity impacts treatment response. She's been defining how tumor heterogeneity contributes to disease progression and therapy resistance. Another area has been the role of breast epithelial progenitor cells in the context of risk of breast cancer, for example, among BRCA1 and BRCA2 affected individuals. Nellie has won a number of awards, including the Paul Marks Prize and the Rosalind Franklin Award for Women in Science, among others. She is a AAAS fellow and is supported by an Outstanding Investigator Award from the NIH. A housekeeping item, please enter your comments or questions in the chat and we'll moderate at the end. So it's great to have Dr. Nellie Polyak here today speaking about response and resistance to BET bromodomain inhibitors in triple-neg. Welcome.
Okay, thank you, Vafik, for this very nice introduction. And Vafik and I have a long history, like, you know, the P21 or VAF1 discovery when I was working on P27, and then we were both postdocs at in Bert's lab many years ago now, it feels like. So today I'd like to tell you about some of our recent results on trying to explore develop combination therapies for triple-negative breast cancer using the bromodomain inhibitors. And if I can have the first slide. So that's my disclosure. Nothing is related to what I'm presenting. And just a brief introduction to some of you who may not be working in a breast cancer area. As you know, breast cancer is not a single disease. There are multiple subtypes that in the clinic we call classify according to the expression of hormone receptors and HER2. And they classified into ER/PR positive endocrine responsive disease and HER2 positive disease that has an amplification of HER2. And then the triple negative disease, which basically means that it's lacking ER/PR and HER2. And currently, this is the only major breast tumor subtype that is lacking targeted therapy. The approved treatment for triple negative disease is still essentially chemotherapy, with more recently combination with PD-L1 checkpoint inhibitors, although just the most recent results from the IMpassion trial seem to be questioning that combination. But, you know, in part due to this lack of targeted therapy, triple negative disease still has fairly poor outcome. Many patients recur very quickly in the first 5 years, and people who don't respond to chemotherapy, they essentially have not many treatment options. There have been, of course, these genome sequencing studies in triple negative breast cancer, and unfortunately, besides p53, p10, RB, and PI3 kinase pathway, almost no other mutations that have been identified that are recurrent and would be observed in multiple tumors, and also that are therapeutically targetable. p53, in fact, is the most commonly mutated gene in triple-negative breast cancer, but as you know, targeting mutant p53 so far has not been successful. Therefore, there are many other approaches needed to more effectively treat triple-negative disease. And epigenetic regulators or epigenetic targets have been an emerging group of proteins that have been explored for therapy in cancer, breast cancer, and other cancer types. And these epigenetic targets can be divided into like writers, for example, DNA methyltransferases or histone methyltransferases that basically put some modification or the DNA or in the histone tail. There are other erasers which remove these modifications, such as histone deacetylases, and now we know that there are even DNA demethylases. And then there is a third group that we call the readers that basically read these epigenetic modifications and then regulate transcription accordingly. So the BET bromodomain proteins belong to this reader subgroup of epigenetic regulators, and BRD4 is one of the prototypes of these BET bromodomain proteins, and JQ1 is one of the prototypes of the inhibitors of these bromodomain proteins. Basically, these bind acetylated histone and regulate transcription that way, and then the BET inhibitors disrupt this interaction with the acetylated histones and inhibit the function of bromodomain proteins that way. So these inhibitors were developed a number of years ago. Jay Bradner was one of the labs that developed JQ1. It's named for Jun Ki, who was a fellow in Jay's lab at that time. And these have, JQ1 has very good specificity in terms of like inhibiting these BET bromodomain family of proteins like BRD4, 2, and 3, and then also some BRDT as well. And they seem to be, there are many other bromodomain proteins, but these seem to be targeting specifically these. And the question is like, how does the specificity of these inhibitors come? So as I mentioned, bromodomain proteins bind acetylated histones, and then they, interact with the transcription machinery like a TFEB complex and regulate transcription that way. And this is what's disrupted by these inhibitors, as you see here. And it's thought to be that in cells, tumor cells, and even normal cells, there are these so-called super enhancers that were described by Rick Young's group, which is basically like a cluster of enhancers around genomic region and genes that seem to be particularly important for the function of the cell in a normal tissue, or in the case of tumor cells, they seem to be particularly important for driving tumorigenesis. For example, MYC is a super enhancer in several cancer types. And then basically we think that, these super enhancers drive oncogenes like MYC or other oncogenes in tumor cells. And then when we're treating with these BET inhibitors, then we basically can have specificity for these cells by disrupting these super enhancers and inhibiting transcription for these super enhancers. So we became interested in the BET bromodomain inhibitors shortly after Jay published the JQ1 paper and also published a follow-up paper showing that MYC is one of the targets of these inhibitors, because MYC is a super enhancer in multiple myeloma and also other cancer types. And the reason is because MYC amplification, which is on 8q23, is one of the most common copy number gains in breast cancer, including triple-negative breast cancer. So first, we actually tested these BET bromodomain inhibitors, multiple different ones, including JQ1, and in a panel of breast cancer cell lines of different subtypes. So the red color indicates the triple-negative cell lines, light blue is the HER2 positive ones, and dark blue is a hormone receptor positive breast cancer cell lines, and red indicates high sensitivity, black indicates lack of, lack of response or resistance. So as you see, like, it's not an absolute correlation, but in general, most triple negative cell lines in here, they're highly sensitive to multiple different BET inhibitors, whereas the luminals in general, again, it's not an absolute for black and white, but in general seem to be less responsive. And similarly, when we downregulate BRD4 or 2 with siRNA or shRNA, we're getting similar effects such as inhibition of growth, and again, preferentially in the triple negative cell lines. So we thought that that looks interesting and potentially a viable new therapeutic strategy for triple negative breast cancer cells. But when we tested these triple negative breast cancer cell lines, we also noticed that the cells fairly rapidly, within a few weeks of culture, develop resistance to these inhibitors. As shown here, this is SUM159 is one of the mesenchymal triple negative breast cancer cells. The parental cells are responsive, and then the resistant cells have a shift in IC50 and essentially keep growing even in high concentration of the drugs. And the same with the SUM149 cell line, which is a more basal-like BRCA1 mutant triple negative cell line. It's actually the parental cells are very effectively inhibited, and the resistant cells even stimulated a little bit at the lower concentration of JQ1, but they definitely don't seem to be responding. So then the question was, what is the mechanism of this resistance and response and resistance to bromodomain inhibitors? And we published a paper a few years ago on these initial findings, and we and others have identified a number of resistance mechanisms. For example, in our case, what we found that in the resistant triple negative breast cancer cell lines, BRD4 is actually stabilized on the chromatin through interacting with MED1, which is part of the mediator complex, and also hyperphosphorylated by CK2 due to the inactivation of PP2A, and therefore they are not responsive to the BET inhibitors because they're binding the chromatin not through the acetylated histone, but through MED1 directly. Therefore, the drugs don't work. So we published this paper, and as a follow-up, we wanted to do a very comprehensive and unbiased profiling and functional characterization of the response and resistance to BET bromodomain inhibitors with the goal of identifying synthetic lethal interactions that we could explore in the clinic, because I mentioned that the tumors very quickly become resistant, and therefore we know that we need combination strategies. So this is a summary of this approach we took, and as you see, we did a very comprehensive molecular profiling of looking at the epigenome, DNA methylation, ChIP-seq for histone modification, and also the bromodomain proteins, as well as histone mass spec, which is a quantitative assessment of histone modifications, and this RPPA, which is looking at the phosphorylation of proteins. We also looked at mutations by whole exome sequencing, RNA-seq for gene expression profiling, then RIME, which is basically looking at interacting proteins on a chromatin. And then we also performed functional assays such as CRISPR screen and small molecule screens in a panel of triple negative cell lines. And we also explored the heterogeneity of the cell populations by performing cellular barcoding and also single cell RNA-seq or ATAC-seq. So I think it's one of the most comprehensive screens for any kind of drug to identify synthetic lethal and resistant interactions. So for the CRISPR screen, we scored the hits. We performed the screen in 4 different cell lines. The 2 parental cell lines, the SUM149 and 59 that were sensitive to JAK1, and also the resistant derivatives that were resistant. And we did the screen in the presence and absence of JAK1. And these plots basically depict the hits that were hits only in the presence of JAK1. The blue ones are hits that basically, if you delete those genes, then the cells are more responsive to JAK1. And the red ones are when they are deleted, then they conferring resistance. And what was very nice, as you see that we did this screen in 4 independent, 4 cell lines, and we got very consistently the same hits, either the same gene or the same pathway. For example, CDK4, BRD2 were the top synthetic lethal hits in all of the cell lines. And similarly, kind of, opposite end, like CDKN1A or Wafix-Weber gene, and no, it's actually P16. And ARID1A and RB were the resistant hits, and BRD7 as well. And we also got many like mediator components, and interestingly, multiple proteasome component proteins like SPOP or CAL3 that could be regulating stability of the bromodomain proteins. The ones that I highlighted in blue, like CDK4, BRD2, and in red, BRD7, were the ones that we ended up following up. CDK4 because of the therapeutic, clinical relevance, as CDK4/6 inhibitors are already in the clinic in breast cancer, not for triple negative, but the hormone receptor positive ones. And BRD2 and 7 we thought were interesting because BRD2 is one of the other bromodomain proteins, whereas BRD7 is part of the SNP5, the BEF complex that has been, as you see, multiple other components of that complex like ARID1A, and also some others are of that resistance. And this again, what we also did is, because we did these screens in a subset of cell lines, and as you know, there is this very useful database of the Broad Institute, the DeepMap, based on a large panel of cancer cell lines of regardless of tumor type. And what this plot shows here, that the genes that were codependencies for BRD4 in that DeepMap screen are also overlapping significantly with the hits what we see, which is kind of reassuring in a way that, totally independently in other lines, like CDK4, for example, comes up as a dependency in the cells that dependent on BRD4 and so on. So that just shows a good agreement among different data sets. We validated these CRISPR hits that we selected by deriving single cell clones and multiple different guides, as you see, confirming that actually when we knock out CDK4, for example, we can make the resistant cells more responsive to JQ1. The same is true for knocking out BRD2. And then BRD7 knockout, on the other hand, makes the cells more resistant to JQ1, validating the CRISPR hit findings. In RPPA, which is basically looking at phosphorylated or proteins or protein level as well, interestingly, many of the pathways that came up, this is again, we did a very comprehensive way, like looking at different time points after JQ1 treatment, 3, 12, and 24 hours. And again, we did in a parental and in a resistant cell lines. And here again, we got a number of pathways that came up as, potentially, involved in mediated by JQ1. But the one that was very consistently, again, a hit is cell cycle regulators, and particularly regulators of G1S transition like RP, and then the CDKs and V1 as well, again, which is nice synergy. RIME, which is to look for our, BRD4 interacting proteins. This is in a SUM149 cell line, again, different concentrations of JQ1, and the parental and resistant cells. We identified a number of proteins that are preferentially binding in the presence of JQ1 and in the resistant cells. So for example, MED1, that, as I mentioned, we previously identified as a resistant mechanism, you see that in the parental cells, BRD4 actually is dissociated from MED1 with JQ1, but in the resistant cells, it's not. It remains associated with MED1 as well. And then again, several of the mediator complex proteins and the protein degradation like SPOP, which is interesting because it's involved in prostate cancer come up as BRD4 associated proteins. Then we also did, in the same 4 cell lines, we did small molecule inhibitor screens, again, using selective library compounds, 144 compounds, and then also an anti-cancer library, which I think it's close to 400 compounds in these indicated pathways. And these plots here just basically depict like we scored each inhibitor in the parental and resistant cell lines in the presence and absence of JQ1. And these top hits here are the hits that are hits in the resistant cell line and in the presence of JQ1. And again, as you see, it was nice to see that the CDK4/6, cell cycle inhibitors come up as top hits, HSP90, and also some microtubule and DNA synthesis agents as well came up in multiple of the cell lines. This is just ranking these inhibitors in the resistant cell lines. And as you see that, confirming that the BET inhibitor resistant cells are resistant as well. But again, like the palbociclib and paclitaxel, which was one of the inhibitors here, was very interesting in terms of being actionable very easily in the clinic. So then again, we validated these hits by performing synergy screens, and CDK4/6 inhibitor palbociclib in these plots, if the dots are below this line, that means synergy. If they are above the line, it would mean antagonism or additive effect. And as you see, this palbociclib is very strongly synergistic in all of these cell lines that we used for the initial screen. Paclitaxel seems to be pretty good synergy, particularly in the resistant cell lines, and also some other like, eribulin and vincristine, which is another microtubule inhibitors, and doxorubicin, gemcitabine, some other chemotherapeutic agents. Again, the resistant cells in particular seem to be more sensitive, and some other, pathway inhibitors that YAPT has, and AXL and SRC kinase inhibitors as well in some cell lines. So again, we followed up palbociclib and paclitaxel in further detail, again, because it's clinically actionable both in breast cancer. So we validated for palbociclib, we validated this synergy in multiple triple-negative breast cancer cell lines that we selected based on differences in RB and p53 status. So some of these are RB mutants, some of them are p53 mutant or wild type. And again, you see that in multiple of them, like PMC, this one, we see very strong, significant synergy. Then, of course, we wanted to validate this finding in xenograft assays, and initially, validated it in the same SUM159 cell line in xenografts. The drugs, JQ1 is the red one, palbociclib is blue. Combination of the two is the pink. And then we also validated taxane, paclitaxel, which is the yellow. And then again, the combination here is green. And this is in a parental SUM149 cell line. We see very strong effect of tumor rate with a combination of palbociclib of JQ1. We do see that palbociclib plus, JQ1 plus paclitaxel also gives a pretty good response, better than JQ1 alone, but is not as good as palbociclib. In the resistant cell line also we see similar effect. And then in a patient-derived xenografts, this IDC50, as you see here, we see a response with JQ1 and palbo alone, but the combination is the more effective. And this just shows here in the MRI imaging that we actually were able to get pretty good shrinkage of these tumors, even relatively short term, like 2 to 3 weeks of treatment of the mice. And then we also wanted to investigate, what is the functional, what is the mechanistic basis of like BRD2 deletion conferring, synthetic lethal interaction, whereas BRD7 deletion conferring, like resistance to beta-bromodomain inhibitors. So first we did ChIP-seq for each one of these proteins and also for the acetylated H3K27 histone. This is in a SUM159 cell line. And you see that they look like virtually indistinguishable. They seem to be binding to the same acetylated histone areas, which is kind of expected because they're supposed to bind acetylated histones. But then what was different about this protein and chromatin binding is when we treated the cells with JQ1. In a parental cell line, you see here, when we plotting the fold difference in binding in a BRD4, BRD2, or BRD7, the most efficient dissociation from the chromatin occurs with BRD4. You see that in the presence of JQ1, you're actually stripping the chromatin from BRD4. BRD2 doesn't dissociate that readily, which could explain why deleting it is actually synthetic lethal with JQ1. And interestingly, BRD7 actually increases binding in the presence of the drug, suggesting that it maybe replaces the BRD4 that's dissociated from the chromatin. In the resistant cell lines, we see the opposite, that BRD4 is actually binding strongest, and BRD7 is actually binds less and a little bit, not significantly, but dissociated from the chromatin. So that was interesting. And then when we're looking at the overlap between these regions that show the increase or decrease in binding, they seem to be actually overlapping, meaning like that the regions that are where BRD4 is coming off the chromatin after JQ1, that's where we see an increased binding for like BRD7 as well. And then we investigated what is the, why are these BRD7 knockout cells are resistant to JQ1. We did gene expression profiling of the BRD7 knockout, multiple clones, and we found that the genes that are differentially expressed between knockout and wild type are very significantly overlapping or enriched in the genes that are significantly different between the parental and resistant cell line in the presence of JQ1. So basically just deleting BRD7 alone makes the cells, switches the profile of the cells to that of these acquired resistant cells. And this on the right-hand side just basically shows that the differentially expressed genes are enriched in the same category between the parental wild type and also BRD7 knockout cells as well. And we saw a very good, we did ATAC-seq for looking at chromatin changes in the BRD7 knockout wild type cells. And we see that in both clones, we see that the genes that are increased in expression, not surprisingly, are the ones where we see an increase open chromatin with ATAC-seq, and then the decrease as well. And the region, the motif that was most significantly enriched for the chromatin that was open in the more open in the BRD7 knockout cells, enriched for the TID motif, which is is the TID is the YAP signaling pathway, again suggesting that maybe the gain of that signaling pathway, which correlates with our CRISPR hit, could be actually what reorganizes the chromatin of these resistance BRD7 knockout cells and make them more resistant. Interestingly, we also observed, we previously have seen that the acquired resistant cells seem to be more luminal. And what we see here, that the BRD7 knockout makes the cells more luminal. This is kind of a luminal mesenchymal score of the SUM159 cell line. So we see a very significant increase in luminal genes and a decrease in the mesenchymal, suggesting again that these cells basically remodeling the chromatin and switching, there's an epigenetic switch to a more luminal state, which is more resistant to these inhibitors. And this also highlights, we saw some particular changes in super enhancers that again overlapped between the BRD7 knockout wild type and the SUM159 parental resistant cells. And I'm highlighting BCL-XL, BCL2L as one of the super enhancers. As you see, it's gained in the resistant cells and also gained here in the BRD7 knockout cells. Again, just suggesting how similar this one knockout it basically makes to those cells. Single cell RNA-seq profiling we performed again in the parental resistant cell lines minus in the presence absence of JQ1. I don't go through much details here. What basically we wanted to see how homogeneous are the populations and to what degree JQ1 shifts the whole population versus not. what you see that for example, in the parental cell lines that are very responsive to JQ1, essentially the whole population shifts. Whereas in the resistant cell lines, we see like multiple subclusters, and they do still shift in the SUM159 line, but not so much, you know, a lesser degree in a SUM149 cell line. And, you know, again, like classifying the cells into like basal, luminal, and also responding, not responding to JQ1. Here, what I just like to highlight that if we classifying this single cell RNA-seq data into JQ1 treated resistant signatures or parental signatures, so there are basically 3 states. In the parental cells, most cells are parental signatures. In the JQ1 treated one, they mostly JQ1 treated signatures, but you do see some cells, even in before treatment in the parental cells, that seem to have the resistant signature, suggesting that you have a pre-existing resistant population, which then gets enriched in the resistant cell line. But interestingly, in a SUM159 cells, a subset of cells still responds to JQ1, because you see like the untreated, treated condition, there is a shift. Whereas in a SUM149 cell, it's much less fraction seems to be responding, which is consistent with what we see the data in terms of like, you know, response of the cell lines. So then we wanted to investigate this further in exploring whether this resistance to the bromodomain inhibitors, is it pre-existing or acquired? And, you know, the single cell data does suggest that there is some acquired resistance because we see a shift, but then also suggests that maybe some pre-existing resistant cells, at least based on these profiles. But to really conclusively address this, we did cellular barcoding. So we basically using this high complexity libraries that we introduced into this SUM159 cell line. And then we cultured the cells in the presence of DMSO, JQ1, or palbociclib, or the combination of the two. And then at different passages, we basically sequenced the cells and looked at the barcode frequencies. And then what Jen, who did this experiment, noticed that when she was passaging the cells, they seem to be slowing down. The combinations seem to be slowing down around passage, you know, 4, 5, and it took much longer to become confluent. The single agents didn't seem to have very significant slowdown, and this indicated like a bottleneck of session that would go through this slowing down phase. And to quantify this from the barcoded data, we calculated the Shannon index of diversity. This is the formula, how you calculate it. And basically, if the diversity is low, that means that you have a few clones that represent most of the cells, and you have an uneven population because you skewed to very few clones. Whereas if your diversity is high, then you have many different clones and you have an even population. So when we barcoding the cells and starting out, we have a high diversity because we use the high complexity library that had about 1 million different barcodes. And then when we culturing the cells, as you see, there is a gradual decrease in barcode diversity, even in a DMSO, because, you know, when we culturing any cell line, there is always some changes. There is more decrease with a single agent like JQ1 or even a little bit with palbociclib alone, but really the most significant drop in diversity, which coincides with the passage number where she noticed this slowing down, is with the combination, which again suggests that you actually have this bottleneck of the cells going through, and only a subset of the clone is able to survive the combination treatment. And then the end of the, if we're looking at which barcodes make up like 50% of the population, in a DMSO, it's many different barcodes, it's a few hundred. In JQ1, it's less than 10. In palbociclib, it's maybe around 100. And then the combination of JQ1 and palbociclib is like below 5. Again, suggesting that combination treatment actually works the best, but you can still get resistant clones, but it seems to be much fewer than single agents alone. And this was actually plotting the data differently, like really following the actual clone frequency. So in this y-axis here is the frequency of clones that have at least 0.5% of all the barcodes. The rest is gray. So for example, you see that in a DMSO control, almost no barcode is really have a 0.5% frequency because there is no selection going on in the parental, in this population. Whereas in the JQ1, the replicates, triplicates actually, you seem to be selecting for the same barcode. So the color here reflects the same clone, the actual same sequence. And this green and dark blue seems to be expanding over time in the JQ1 treated population. In the palbociclib alone, we see some. And then in the combination treatment, we see the same dark blue and green, and then a few other clones seem to be expanding as well. Again, suggesting that there is a selection going on for these pre-existing populations with this particular barcode frequency. And I don't go through the math, but Jen did mathematical modeling to predict what is the frequency of pre-existing and acquired resistant to JQ1 or palbociclib. And as you see, it's actually pretty high. The acquired resistance in 1 in 20 cell division, which is very high, considering, these numbers, and even the pre-existing resistance seems to be fairly high for palbociclib. So no wonder why single agent treatments never really work, especially not in advanced stage disease, because your probability of getting a resistance is very high. Then Jen also investigated what is the mechanism of resistance to the combination agents, JQ1 and palbociclib, and also, taxanes, but we mainly focusing on palbociclib. So she took the cells that were cultured, these barcoded cells that were cultured over time, and did mutation analysis, gene expression, and also single cell profiling. And as you know, palbociclib, it inhibits the CDK4/6 cyclin D complexes that phosphorylates RB. And loss of RB is one of the known resistance mechanisms to palbociclib treatment. And indeed, in our cells also, we found this RB mutation that basically would change RB, and it converts to a nonsense mutation. So it basically would be a truncated RB, suggesting that this could be contributing to the resistance. And using droplet digital PCR, we actually found that this clone was present in a pre-treatment population at very low frequency, like 0.001% of the clones. And then as you see with the combination treatment in particular, but even with palbociclib, we see some enrichment. Interestingly, it never became 100% clone, but it definitely went up to like almost 10%, or in one of the replicates about 30%. So it's a very significant enrichment for this clone, but it's not the whole thing because it was not homogeneous as well. And just to prove that RB loss is actually conferring resistance to JQ1 palbociclib combination, we deleted RB in this cell line and show that these cells that have the deletion, they are not responsive to the combination of JQ1 plus palbociclib compared to the wild type cells that are effectively inhibited. And other ways plotting this, the synergy that we see in the parental cells, we don't see in the RB knockout cells. So it's certainly one of the mechanism of resistance. Based on RNA-seq of these resistant cells, we also found that the cell cycle regulators, many of them, like for example, cyclin E, we see a significant upregulation, and many other cell cycle regulators seem to be overexpressed. That again could contribute to resistance to the combination treatment. But what was interesting when we looked at the single cell RNA-seq data, and this is the t-SNP plot of the data, this is just putting, highlighting some genes that seem to be dividing the clusters. In the case of JQ1 plus palbociclib resistant cells, you see there are 4 very distinct clusters of genes with different expression pattern. We see some genes that seem to be more mesenchymal features. We see some cluster that's more cell cycle regulators, again, highly expressed. This is looking at the pathways that seem to be enriched in these. So that basically highlights that even though we have a resistant population and a subset of these cells were grown out of the acquired, pre-existing resistant clones, there is still a heterogeneity, and there are multiple different ways these cells can become resistant, even to combination agents such as JQ1 plus palbociclib. And one of the interesting observations we had when Jen, I like looking at pathology slides, and when Jen was collecting the xenografts from those validation studies, we noticed that you see these H&E images that the cells look in these palbociclib plus JQ1 treated, but even palbo alone, and a little bit with a taxane combination. You see these gigantic, really big cells with multiple nuclei, which is kind of suspicious of something going on with ploidy of the cells. So when we actually decided to do karyotyping of this cell line, so this is the SUM159 parental cell line. It's an unusual cell line. It's almost completely diploid, the parental cells, maybe because it has a POL-E mutation, so it probably has a highly mutagenic and remains chromosomally stable. And then, surprisingly, the JQ1 resistant or palbociclib resistant or combination resistant cells seem to have an almost complete genome doubling. So they became almost completely tetraploid during treatment, which we see in vivo in the xenografts and also in cell culture. So we wanted to investigate this further because that's kind of interesting, like, tetraploidization leads to resistance. And to really explore this, we did FACS, and then we also labeled the cells with FTSE, so that way we could, using the FITSU system, we could actually differentiate the cells that are G1 cells, but they are tetraploid versus real G2M cells that are normally diploid. And as you see here with these palbociclib JQ1 combination, we saw that the cells became nearly or completely tetraploid after treatment, and this correlates with the karyotype. And this actually seemed to be happening fairly quickly because Jen did a time course, and within 7 days of treatment, we already see a very significant shift to the tetraploidization, even with a single agent, but particularly with a combination. And then you see here with the staining with the tubulin, these cells, you see that they are these multinucleated cells that particularly arise in the presence of the combination treatment. And this is seen not just in a SUM159 cell line, but actually multiple cell lines that we tested. In even as MCF10A cells, which is not a cancer cell line, but it's a basal cell line, we see very effective tetraploidization. And it was somewhat surprising because this cell supposedly have wild type RB, and polyploidization is supposed to be, in the presence of wild type not RB also, and p53, they're supposed to be not surviving, but they seem to be surviving. And we also see this with the other agent like ribociclib we tested and abemaciclib. So it doesn't seem to be specific for the palbociclib, and it doesn't seem to be specific to the cell line, but we see it overall. And then we wanted to see, like, why does it happen? Like, what do their cells, why they cannot complete mitosis and seem to become multinucleated? So Jen derived the cells that have expressed GFP histone and then this tubulin in red, so we can do live microscopy. And you see in the normal conditions, this is a normal mitosis, very pretty, the cells condense and then divide, split up the nuclei, and then they separate. So that's a normal mitosis. In contrast, in the palbociclib plus JQ1 treated cells, we saw many aberrant mitosis when the cells somehow cannot complete cytokinesis, so they end up with multinucleated cells, or they cannot even really complete mitosis, and they seem to be re-entering G1 without ever really dividing up, like this cell here. You see, and then seem to be going back, not dividing, and seem to be just continuing to grow. So that looked like there seemed to be many different mitotic abnormalities in these cells. We actually quantified it, looking at, with the help of David Pellman's lab, which phases of the interphase or prometaphase, metaphase, anaphase we see. And as you see that the combination treated cells, but even in a single one, we see many arrested mitotic cells that are not able to really complete mitosis, and they seem to be re-entering the cell cycle phase. And we investigated that the tetraploid cells by themselves actually not immediately resistant to the drugs. I don't show it here, you can look it up in the paper, but the tetraploidization seemed to be accelerating the evolution of drug-resistant disease. And there have been, as you know, a number of studies showing that in human tumors, the ones that seem to undergo whole genome doubling, like Charlie Swanton is one of the labs who investigated this area, they seem to have worse outcome. And in our case, what we're suggesting that this tetraploidization basically accelerates the evolution because it seems to be maybe contributing to increased heterogeneity of the population. So just to summarize our findings, what we see that there are multiple different resistance mechanisms to single agent and even combinations. There seem to be both pre-existing and acquired. The acquired resistance is several hundredfold more common. There seem to be a G1S regulator seem to be critical for the response to JQ1 and palbociclib, and we see this tetraploidization happening fairly quickly. And the combination, I again didn't show you, but we actually also tested what's better to apply combination therapy early on or use single agent, which is very commonly done in a clinic, that you're doing one agent and they're resistant and you give another agent. But it's probably combination starting from the beginning would be the best. And one of the somewhat unexpected finding was that triple negative cell lines, even the ones that have RB and P53 mutations, seem to be sensitive to CDK4/6 inhibitors. And one mechanism, again, I didn't get into details, but CDK4 actually regulates the phosphorylation of SPOP, and that regulates the stability of BRD4 and BRD2. So there could be an RB independent mechanism of action, and that could explain why even the RB null cell lines are responsive. And this combination seemed to be very effective in a subset of triple negative. And overall, we feel like we developed a number of combinations. The two that is most actionable clinically, and we already designed a clinical trial, is a combination with palbociclib plus JQ1. JQ1 is not in the clinic, but there are BET inhibitors that are in the clinic. And the other one is paclitaxel combination, because paclitaxel is already used and approved in the clinic, even for metastatic triple negative breast cancer. And we also have been testing adding PD-L1 on top of this combination. So basically having triple agent, and we have preclinical data, again, suggesting that's even more effective treatment. And we have the clinical trials that we hoping to start in the next few months after we get approval. Everything has been unfortunately slowed down in the past few months, but hopefully we can actually test this in some patients. And lastly, I'd just like to acknowledge the people who did the work. I already mentioned Jen, who's a very talented MD PhD student. This was her PhD work, now finishing up in a clinic, and she's actually a Brown alumni. She did her undergrad at Brown. Xiaokun Xu is a postdoc who now got a position at Beijing University, who did the initial CRISPR screens and those studies with the help of many others in the lab. And then we have been collaborating with Francisca Mikor's lab, who is a computational biologist, and Hua Jun, who is a postdoc in her lab, who particularly helped with the informatics. And we also have collaborators at many other institutions, as well as the Ludwig Center at Harvard. John Brugge and Isaac Harris have helped with the small molecule inhibitor screen. And thank you for your attention, and I'm happy to answer any questions you may have.
Thank you very much, Nelly, for that nice talk. Please put your questions in the chat, and maybe I'll start with a question about the tetraploid mechanism. Do those cells actually proliferate at some point? I mean, can they grow in a tumor, or do they just sit there, stay alive, but aren't really growing as a tumor?
Yeah, so they seem to be growing, because we can actually passage those cells, and they are growing in the presence of the combination agents for several, weeks, we did at least. And we also had the xenografts. That's why we kind of actually started this whole ploidy study, because we noticed in the xenografts that there are these gigantic cells in the mice that were treated with a combination. So they are able to form tumors. They are slower growing, but they still, growing. We kind of be following this up and trying to figure out, how does it happen? And also, how come they continue to grow? Because, they have this mitotic abnormality, and they still see able to re-enter the cell cycle and proliferate. And we even see some 8N and 16N cells at much lower frequency. So they seem to become, genome doubling, at least at some frequency.
And do you think there's some sort of a gene dosage effect with, the extra chromosomes, or it's just the mitotic problem that makes them resistant?
I think what we are thinking, and this is what we're exploring, that whole genome doubling and tetraploidization basically increases the heterogeneity of the population, because it kind of makes increases transcriptional heterogeneity and also increases cell-to-cell heterogeneity in a way that, the cells can more have many different phenotypes. And that's how we could accelerate evolution. And, like in a real human data sets like TCGA, you can actually identify tumors where you clearly see that there was a whole genome doubling based on, seeing everything duplicated. And those tumors tend to do worse. They're more likely to be metastatic and resistant to treatment. And it's not just the of course, you can argue that there is something that mutated or something enables those cells to survive, in a tetraploid state that could directly contribute to resistance. But at least in our case, it looks like it's not just the tetraploidization itself is not immediately making them resistant. It's just enabling rapid evolution of resistance.
Okay, we have a question from Botai Swan. Hi, Dr. Polyak. Very interesting talk. Just to add to that question, my project in the Dawson lab involves studying a subpopulation of polyploid breast cancer cells in triple-negative breast cancer. We find that these cells can proliferate amitotically through budding. Have you observed this phenomenon?
Oh, budding. We haven't even looked for that. Like budding, like the budding yeast, I guess. We can look, because we have these live cell imaging studies. I guess that would show us if we see that budding, at least in those conditions. But no, we haven't looked at it. So that's an interesting idea. Yeah.
We have a question from Dr. Doug Anthony. Excellent talk. Thank you. Have you looked at other cancers? Are these pathways limited to triple-negative breast cancers?
We have not looked at other cancers because, we're working on breast, but other labs have seen that, serous, high-grade serous ovarian cancer, which in many respects is very similar to triple-negative breast cancer, seem to have very similar response to BET bromodomain inhibitors, and also prostate cancer, particularly androgen-resistant prostate cancer. And there's some clinical trial, the company that we also, exploring, there's a clinical trial going on by Zenith that basically tests these combination agents with BET inhibitors in prostate cancer. So it certainly can happen in other solid tumor types, we have been focusing on triple negative breast. We also see that, luminal breast cancer could respond because, palbociclib has been approved for the treatment of hormone receptor positive tumors and in combination with endocrine therapy. And there is some data suggesting that if you're adding JQ1 on top of it, then you get even better response. So it certainly can happen different subsets of breast cancer and also outside of breast cancer. Ovarian and prostate are the two that I know of seem to be behaving similarly.
Here's a question from Dr. Mahendra Singh at University of Miami. Great talk, given MYC turned out to be one of the major genes in synthetic lethality screen, and aurora kinase inhibitors have to be shown to be affecting aurora kinase MYC signaling axis. They're wondering how would these cells, especially the resistant ones, respond to combination of aurora kinase inhibitor and other agents that were tried in this work.
So we didn't follow up those hits, but when I showed the small molecule screen results, and again, you can look up the paper, aurora kinase inhibitors, did come up as one of the agents that synergize with JQ1. Also, the resistance cells were more sensitive to it. So it certainly could be explored. We haven't done it. HSP90 inhibitors is another one. We did some follow-up, and JAK inhibitors. The one that looks very interesting to me is the, AXL and YAB-TES pathway, because this seems to be coming up in multiple different resistance mechanisms, not just BET inhibitors, but other drugs in other agents. And then the other one that's very interesting, I mean, it's not easily druggable, but, the SNF5 complex, the ARID mutation is one of the most common mutation in metastatic treatment-resistant breast cancer, suggesting that it can contribute to resistance, and it came up in our study as well, that complex. So there's a lot more than what, you can follow up. I mean, we deposited all the data, and hopefully it can be used for others to create other interesting stories, and hopefully clinical trials that could be tried.
We have a follow-up question about the polyploid cancer cells. Thank you for the answer. We have found these cells can give rise to daughter cells by splitting off their nuclei to give rise to fully viable proliferating daughter cells. We've also found distinct abnormalities in the expression of cell cycle genes that you have also identified in your work. I would love to follow up on this through email after the talk.
Okay. Just send me an email. I think you can find me, Google. Yes. I mean, we're very interested in this ploidy because of the, known clinical importance, and also for this evolution mechanisms, what we think,
There's a question from Zizan Gu. Recently, we did a tissue array on 415 breast patient samples in Shiga Medical University Hospital, Japan, and RNA-seq array on 336 triple-negative breast cancer patient samples at Fudan University Cancer Center, Shanghai. All patient samples were from 2012 till 2020. Surprisingly, in both hospitals, we have observed that TNBC 5-year survival rate is already above 92 to 94%. What do you envision on the TNBC research field? Are we done with the job already with the new Roche PD-L1 drug?
So that survival, first of all, survival is very much dependent on stage of diagnosis. So if you like a stage 1 disease, the survival can be, I don't think it's that high, but it can be, relatively high fraction of patients survive, maybe closer to 80%. But then if you have a more advanced stage disease, even stage 3, then, and stage 4 survival is below 25%. So I think, I don't think you are done, and I would never say we are done, because even the hormone receptor positive disease that, you know, the 5-year survival is very good if you diagnosed early, but then you can have late recurrences. So for triple negative, that's a paradox, that you have very few late recurrences. Pretty much everybody who dies and doesn't respond happens very steep drop in the first 5 years, but particularly the first 2 years. And I think that's because probably many patients are already metastatic at the time of diagnosis. And then the PD-L1 drug, if you've seen the most recent FDA warning, as recent as yesterday or a few days ago, there was this promising trial of the, you know, combining paclitaxel with PD-L1 in patients who are PD-L1 positive, which is only about 10-15% of triple negative patients. And those had some, you know, survival benefit by, you know, 6 to 9 months. And that was a type of form of paclitaxel that is conjugated with the albumin, so it has a better PK profile. Now they just published the data on the IMPassion trial, which is using regular paclitaxel, and then basically the patient's actually doing worse with PD-L1 than without PD-L1. So I think, you know, with this immunotherapy, I know there is a lot of excitement. We ourselves are planning a combination trial. But first of all, the fraction of patients that are actually PD-L1 positive, which so far have been indicative of who will likely to benefit, seems to be fairly low. And then also, even within that group, not everybody responds. And of course, there are trials now to do earlier treatment. Don't wait until you have metastatic disease. So I think there is still a lot of work to be done. And, you know, triple negative, the ethnicity and the age of diagnosis could also influence it. I mean, again, for this 94% survival, I'd like to see the stage and the age of that population, because unfortunately here, even at the Farber, we have many young women, and particularly African-American women, who do very poorly, and they don't, you know, they still die of the disease. So I think we still have work to do. And, you know, if we cure everybody, I'll be the happiest person, and I move on to do something else.
We have a question from Dr. Attila Sehan. I might have missed this, but did you validate the single cell synthetic lethality phenotypes in patient tumor specimens to see what type of codependency of these mutations might be leading to drug sensitivity or resistance?
We have not looked at patient tumor samples. That's something that we have ongoing now. We're doing some single cell RNA-seq studies in triple negative breast cancer patients. And then, of course, we're trying to start this combination trial in patients, and then hoping to be able to do correlative studies from people on treatment, because that would be the most interesting and, you know, the best validation. And hopefully we get this off the ground in a couple of months.
Question from Shui Zhao. Great talk. Have you looked at any metabolic change in the resistant cells and after combination treatment?
I think we have not, but, you know, I would not be surprised if we see metabolic changes. We have done it in other projects, and the resistant cells, many times they seem to be different regulation of ROS, like glutathione systems seem to be changing. In this particular cells, we haven't done. I mean, it could be done, but we have not tested.
Okay, well, it's been suggested that we all go ahead and unmute ourselves so we can clap and thank you for the great talk and all the answers to these questions. So
okay.
Why doesn't everybody unmute themselves? I don't have a way to easily do that.
Okay, thank you so much.
Thank you. Thank you.
Thank you. And, you know, it's nice to have these seminars, so we still feel like we connected, even though we isolated, not far apart though, but, you know, we are. So, okay, thank you, Wafi.
Yeah, thank you very much. Have
you.
a great afternoon, everybody.